enough number. So hello everyone, welcome to the Siddharth Tech Talk series. For today's meeting seminar, we are having Dr. Thang Pham, who is from the INKA Proteomics Laboratory of Amsterdam University Medical Center. And Dr. Thang Pham is going to talk about machine learning in cancer proteomics. I hand it over to you.
Yes. Thanks, Gwen. Good morning, everybody. Thank you very much for having me. So I can already answer the first question of Gwen. So we are located here in this red and green building, and it's in Amsterdam, and it's basically part of University Hospital. So my name is Thang Pham, and I'm a computer scientist in the lab. And today I'll talk about machine learning in cancer proteomics. And so if you are a bit outside the field, I think you will be hearing a lot about genomics. And in that sense, proteomics can be seen as like part of genomics studies in the last 10-20 years. But in the field, it's very different from genomics. We study proteins, and there are many processes downstream of the genome that affect the cancer proteomes. And in particular, many molecular targeted cancer drugs are working in this protein space. So it's very relevant. So the technologies that we so the technology that we'll be using is it's called mass spectrometry, and this is a little bit outdated figure, but it gives a very good overview of the technologies that we're using. So in the first step in the lab, we have protein mixtures, and these are digested into peptides, and it can go to 1 or 2 or 3, even 3 steps of separation before it goes to the mass spec. And in the second steps, the peptides, selected peptide will go through the mass spectrometry and break down further. And at the end of the process, we obtain a mass spectrum. And then using the computer analysis, we can match the theoretical mass spectra against the experimental mass spectrum obtained, and then to identify the peptide. So the protein, it's a change of amino acid sequence, and they are typically quite big for mass spectrometry. So the first step people do is to cut it into smaller pieces, and these are typically using trypsin that cut the K and the R, and these peptide will go through the mass spectrometer, and some will fly and some will not. But the ones that are flying in the mass spec here will be broken down into smaller pieces. And the mass spectrometry will measure the mass of each ion. And in this figure, you see that by having the mass of each of these broken smaller pieces and matching to a theoretical mass, a computer algorithm will identify which one is which. So it's quite a lot of bioinformatics before you can get down to machine learning. And in the lab, we do a lot of we are in the medical oncology department where we do a lot of cancer study. As you see listed here, we do colorectal cancer, breast cancer, lung cancer, PDAC, AML, DLL, prostate cancer, and also we do quite a few pan cancer analysis. And the goal is to the two main goal is to look into early non-invasive detection of cancer and also personalized cancer therapy. So in the I mentioned mass spectrometry, and the last 15 years, the data-dependent acquisition has been very popular, has been the main driving force in the field. But recently, there's also a new technique. It's called data-independent acquisition. That is it's an alternative that gives much more consistent data. So the figure here illustrates the two different techniques. So on the left, you see the old technique where the high abundant peptide will be selected and sequenced separately, whereas the new technique would sequence the whole windows, which contains multiple peptides, which makes the MS analysis a little bit more difficult. But then it gives a much more consistent data set. And if you do the machine learning, missing value is a very big problem. And with the new acquisition, it gives much less missing value. Before going to machine learning, I want to give a few slides on bioinformatics because the new technology we have, people have much focusing on identification problem, but to do analysis, downstream analysis, what we need is a protein level quantification. So this figure shows you what one can observe for a single protein that have multiple peptides. And so each, the x-axis here, so the sample, and each peptide has multiple level of intensity, for example. So we want to combine these multiple observations into single protein level quantification. And so there's several methods doing that. One could look at the top peptide or look at the average of all the observation. And there's also a method that's very popular called median polish. But in the field, this MaxLFQ has been very useful. And it was devised for DDA-MS, so that's the previous, well, the method that I mentioned before, that the major technology. And it goes as follows. So if you have two samples, I and J, for every pairwise sample, you can look at the ions that are detected in both samples. So you can take the ratio or the difference in abundance of these two samples. And here there are ions that are missing. So missing values are very frequently happened in mass spectrometry proteomics. And by looking at the pairwise, we can really utilize all the detection points and then brings a comparison between two samples into sort of graph and turn the whole protein quantification into a optimization problem. Can be solved very effectively. And we've shown that this technique can also be applied for the new mass spectrometry technique. And so last year we published a paper in bioinformatics where we also study in detail these the more theoretical property when the peptide can be validly quantified. Basically, if the graph of pairwise sample are disconnected, then there will be no solution for this optimization problem. And we shown on several data sets that this quantification really give better than results than other methods. And this is a slide to advertise our work a little bit. So if you work with the data, with mass spectrometry data, you might want to look up this R package called IQ, stand for ion quantification, and the one of the features that we work really hard on is to is for the software to handle very big data set. So that's just briefly on bioinformatics and proteomics. And now we have let's say a rectangle table of sample of protein. And machine learning, as many of you know, has a very wide application. It goes from early detection to treatment outcome prediction, biomarker discovery, and cancer subtyping, and many more. And I'm kind of quite proud to say that we are actually involved in all of these application field listed here. This is one example, one major study we performed a couple of years ago. So in the Netherlands, for senior people from 65, yeah, there's a test called the fit test that that aim to detect early sign of colorectal cancer. So if the fit test is positive, you will be recommended to go to colonoscopy, which is a very invasive technique. So the aim is to complement this fit test with cancer proteins that can give higher specificity and higher performance. And now you can start seeing a little bit of machine learning techniques. And here actually we use quite standard method. So from a set of, we have two series of a smaller one of 22 sample and a bigger one of almost 300 sample. And with statistical analysis, we zoomed out to 29 markers, and using logistic regression and CART analysis, we came up with 4 protein marker panels. I'll go back to feature selection a bit later, that performed really well. And these 4 protein not only can detect cancer from control, but they can also, we also look at detect very early stage of cancer development. So AI stands for advanced adenoma. And recently, we also perform a big validation of almost 800 sample, but the technology we did not do that using mass spectrometry, but using a platform called ELISA, which is very which is available in in the hospital in most places. So that just give you a flavor, an example of a study that we did. Now I want to go back a long time ago where a lot of this started. And it's one of the first work in genomics in breast cancer from the group of Peru. And here they used unsupervised clustering that make 5 clusters of breast cancer patient. And for each of these cluster, a centroid is calculated. And the classification is using nearest centroid classifier. And if you're into machine learning, this is one of the first technique that one would learn, I guess. It's very simple, but very interesting to see that these cluster of patients do have different characteristics. So this is a Kaplan-Meier plot. So on the x-axis, you see the overall survival months, and on the y-axis are the probability of survival. So here you can see that the lumino A cluster and normalized cluster are having a very good survival rate, whereas HER2 positive or basal group are not so well. So that was genomics, and a few years later, there was study in proteomics. And while this is, as you see, quite almost 15 years old, the problem is still very much the same, and that is you have here it's lung cancer patient treated with EGFR inhibitors, but to do the genomics analysis, they require tumor tissue, which is very invasive. So the aim is whether we can use pre-treatment serum, so the blood sample, that identify a patient that would respond well to this particular treatment. And the way they did that is using a k-nearest neighbor of 8 peptides. So again, if you're into machine learning, immediately recognize that these are very simple machine learning methods. And the algorithm is validated in a blinded validation on 2 Italian cohort. And the result is very impressive. And then it was stated that this algorithm is not only prognostic, so looking at survival, but really can predict patient that respond to this EGFR treatment. So motivated by that, we also perform a study in our lab. We have a cohort of lung cancer patients with 27 patients. And we also doing taking sample at pre-treatment, but also at the end of treatment and during this cycle. And these are characteristics of the patient. And I will not go into detail about this, but for feature selection, we just use a filtering method where we select top peptides using a statistical test, and then we use support of the machine, lead one out cross validation. And these are the results. We have a 6 peptide ion that have predictive accuracy of 82%, and a longer version with 13 peptides that have slightly higher accuracy. And these peptides are used by are derived from time course analysis. And on training data, it performed really well, as you can see here for the 6 peptide signatures, and this is for 13 peptide signatures. But because of the feature selection, where we do statistical tests before the subcontractor machine analysis, the results the results, of course, need to be validated, and we were very fortunate that 3 years later there was this another cohort, a patient of the same characteristic, and we want to look at this cohort of patient. It's a completely independent data set, as you see, 3 years apart. And these are the characteristics of this patient, second set of patient. And in this set, we also have genomic analysis done on this set of patient with EGFR mutation status and KRAS mutation status. So if for people who are not very familiar with this, these are 2 mutations that are very frequently happen in this cancer, and they they drive the cancer, and there's a lot of treatment associated with these 2 mutation. And I must say that this is the 10-year-old study. Now there's a lot more mutation analysis. So these EGFRs is not significant with respect to the progression-free survival and the overall survival analysis, as you see here from these 2 curves. And the KRAS is also not significant. It's the p-value for progression-free survival is low, but not really impressive. So when we apply the subcontractor machine with 6 peptide on this set, we see a very significant differences of the 2 groups. And looking into the mass spec-based predictors, we see that they are independent from the EGFR mutation status. That means that we can use this mass spec results in combination with the genomic status results. So if you look at we look at the cohort of EGFR of patient without this mutation, EGFR negative, we can see that the subcontractor machine can separate the subset of patients who predicted long and predicted short. So that results really interesting because EGFR positive patients can receive this treatment, but the EGFR negative patient, some of them might still be the drug might still be beneficial for them. And we still have a number of ongoing studies in this type of analysis, that lung cancer respond to new chemotherapy. We want to predict early detection for prostate cancer. For colorectal cancer, we have many studies in colorectal cancer, and one of them is a big study where people in genomic already separate, just like the breast cancer analysis. There are several subtyping schemes, but one is called the CMS subtype, where CMS4 would not be benefit. Patient in CMS4 would not benefit from chemotherapy, and we want to identify this subset of patients. And we also want to see if protein data can give a different subtyping. And we also looking at not only cancer, but also other neurodegenerative disease, so on. So I mentioned feature selection, and if you're working in machine learning, I think this is still very much an unsolved problem. So if you're working in this area, I think there's still a lot to be done. So how do we select features? If you look at the literature, you can roughly divide it into three approach. The first one is called filtering, where we just look at the top protein, ranked either by p-value or area under the curve. The problem with this is that many of these top features are correlated, so they are involved in the same processes and so on. So if you try to combine them together, it will not improve the results so much. There are a large class of methods in statistical machine learning that mathematically very elegant. So Pham it's a nearest neighbor with sparse nearest neighbor, so to say. You have lasso, elastic net, and stepwise classifier. So I just make some plot of these classifiers here. One problem with this is that, well, you have very nice, elegant mathematics, but then it has one solution. And one cannot really incorporate expert knowledge very easily. So that's my personal opinion. Except for stepwise classifier or boosting type method, that my personal favorite, because it to some extent give you this flexibility of putting in prior knowledge by the way of designing your base classifier. And there's another approach, the random forest, except next to the classification, of course, it gives you a feature relevance or feature importance, that looking at the rank of these features, here I do not give any names, but these are the number of the frequency that certain protein or genes happen in a good classifier. And it gives you the opportunity to work with domain knowledge expert, domain expert to find good combination, and it's just very important. But at the moment, I think there's a systematic way of doing this is still lacking. I guess you might wonder about the brute force approach. So if you look at, if we look at a study where we want to explore all the combination of 7 proteins that well perform, and by well perform we mean in terms of error rate, it would be in the top 5% lowest, the lowest 5%. So if we just do that brute force with 7 gene signatures of 200 genes, it would be impossible. So it's just a matter of random sampling. And after 1 million trials, so this one to predict actually the BRCA in breast cancer, we can see one of the genes that are top ranking validated in literature. So it's really nice. So it's it's a hypothesis generation kind of approach. And of course, we always ask ourselves, how does it perform in comparison to just random? So we also generate a lot of random orderings, and then we see that with 7 protein, using this approach of top ranking, it performed quite well. So that was feature selection. The next challenge I would like to mention is called small sample size. And with small sample size, one need to incorporate a lot of prior knowledge. And a systematic way of doing that is still lacking at the moment. So this is a study where we look at phosphoproteomics for kinase inhibitor selection based on a single patient sample. And phosphoproteomics, in phosphoproteomics you have kinases and substrates. And the kinase-substrate relations are very important because their interaction basically drives a lot of the signaling process in the cell. So in phosphoproteomics, we look at the phosphopeptide of the kinases itself, but also looking at the phosphosite, and that are the peptide that are activated by these kinases. So for phosphopeptide, we can have just the measurement on the kinase, the kinome, but we also look at the part that are really important. So if the phosphosite is happening in this small, is within a predefined change of amino acid called activation loop, then it should get the importance should be increased. And also we look at phosphosite that from a database that is having a experimentally validated database. It's called the Phosphosite Plus database. And also from a database called the networking database that contain predicted kinase substrate relationship. So I've mentioned a lot of about databases and activation loop. These are the knowledge that that as a computer scientist or machine learner practitioner, it's not easy to come by, and so it's a study where you do need a lot of different expertise, and how to combine all this information into numbers that are easily to work with. So we devise this formula, which is basically the geometric average of the two sides, so the kinase size and the phosphosite size, and then we do a lot of permutation analysis to validate our results. And this tool is available in website called inkascore.org, that people having phosphoproteomic data can use. And we've validated this method or this formula in many different cell lines and preclinical model, and listed here some of the cell lines that having a known driver mutation. So have this cell line, for example, have an ALK mutation, and from the INKA analysis, you see that all these driver mutations ended up very high in the INKA score list. And we also provide a visualization so that domain experts can explore this data set and explore the results. For example, sometimes it's quite visible that a single drug is sufficient to suppress or this tumor, but sometimes a single drug is probably not enough because of the many different hotspots can be seen here. So can we use this profile to predict which kinase inhibitors works? So we look at the data in a Nature publication a few years ago from a group in Italy, and here you have a lot of PDX model. So this is human tissues that grow in mouse, and for many of them, the color ones, you can see that they have studied the driver, the genomic driver that lead to this resistance to the drug. But there are many gray bar here that says no clear genomic operation. And the question is whether we can use phosphoproteomics data to answer this important question of finding out the resistance mechanism. So we took 2 of those PDX model and look at the INKA score of the model and also the organoid that derive, and look at the 3 drugs that inhibit this kinase that have very high INKA score, and validated in the lab that it really works. So next to the phosphoproteomics data, we now have also drug data. So the question is also how to incorporate all this information into the machine learning model. So I'm switching to a more recent work, and I think many of you would be very interested in to know about how where we are with more recent development in machine learning called deep learning. And one of the differences, of course, that deep learning have grown tremendously the last decade, and there's so many software and tools available. And one of the methods, it's one of the method in natural language processing is that you can handle very easily now the variable length input. And 2 years ago, there's a group in Germany that published in Nature method a technique called PROSIT, and they look at the peptide sequence and predict these mass spectrometry properties. So what it would look like if it went through the mass spectrometry. It's very nice. And not only the mass spec, but other separation methods that can be predicted just based on the sequence alone. And the nice thing about this study is that at this level of data pre-processing, we do have a lot of samples. So in total, this study has used 20 million spectra. So you have 20 million of these peptides and the corresponding experimental mass spectrometry output. And in our lab, we also have this kind of data in large number because we are a data generation lab. So But before that, we look at the motivation why we would want to predict the mass spectrometry property. And I go back to this figure here that I showed earlier. And the difference between the two, the existing data-dependent acquisition, the popular method, and the upcoming data-independent acquisition. And the difference is, of course, in the way that because of the acquisition, the MS spectra of one is quite, well, it's not easy, but simpler to analyze, whereas the new method is a convolution of multiple peptides. And it would help greatly if we have a spectral library of known peptide. So this the predicted library would help in identify or deconvolute this set of peptide into single ones. It also has, I show here another work from the group of design, where the prediction of retention time also improve the quality control of your study, especially if you're looking at exotic things like neuroantigen and other non-standard analysis. So we are quite strong in phosphoproteomics. We have a lot of phosphoproteomics data, partly because we are in a cancer center and we have a lot of phosphoproteomics study, and we want to contribute this set of data into litteratures, and we are working on a deep learning model where existing methods have been using this recurrent network model, and we want to implement a transformer model with phosphoproteomic data. So phosphoproteomic meaning that you have extra phosphorylation on certain amino acid. Yeah, and transformer architecture. Some of you might have heard. It's have been very successful in natural language processing, and recently they have been used successfully in 3D modeling, protein folding, and so on. So that come to my last slide, and I just want to conclude with a little summary. So I think I show you a little bit of the field, and the applications are highly relevant, changing from invasive technique, genomic sequencing, to if one could do the same thing if we but just using the blood sample, that would be much desired. It's a multidisciplinary field where bioinformatics play important roles, but also other fields that contribute. And I have said multiple times during the talk where we do need expert knowledge from biology and chemistry and so on. As for machine learning, systematic feature selections and incorporation of knowledge, it's very challenging. I would love to hear from the audience if this is not the case, because then you can just go home and try out your method. I mentioned kinase and substrate interactions, and that's also very important, and that's also their opportunity to apply advanced machine learning to learn more about kinase and substrate relations and how to put them all together for single patient analysis. Is very challenging, and we need to do that because we have only very limited number of samples. and also exciting development in deep learning of modeling protein sequence. Yes, so that concludes my talk. Thank you very much.